Good morning once again. My name is Cynthia Perez. I am a professor at the University of Puerto Rico School of Public Health, and I would like to welcome all of you to the second and final day of the Research on Cancer Epidemiology Conference sponsored by the University of Puerto Rico and the MD Anderson Cancer Center. We hope that day two will manage to stay in line with the previous successful day one, where several researchers share information about social determinants of cancer, studies addressing cancer burden in Latin American and Caribbean countries, cancer survival, and cancer disparities. Today, we bring together several speakers that will stimulate the discussion around the following themes: effect of genetic ancestry on cancer susceptibility, HPV-related cancers, and the palliative cancer care in Latin America and the Caribbean. Our first speaker, Dr. Juan Martínez-Cruzado, is a full professor of the Department of Biology at the University of Puerto Rico Mayaüez campus. He is well known for his pioneering work on the Amerindian ancestry of most mitochondrial DNA molecules in the Puerto Rican population. His work has been recognized as highly meritorious by the Puerto Rico Academy of Arts and Sciences and the Caribbean Division of the American Association for the Advancement of Science dedicated the 2011 annual meeting to him. Dr. Martínez-Cruzado is also an advisor to the Smithsonian National Museum of the American Indian and is part of the 1000 Genomes Consortium. Our speaker will discuss today the ancestry distribution of cancer polymorphisms in Puerto Rico. Please join me in welcoming Dr. Martínez-Cruzado.
Thank you, Cynthia. Welcome, everybody. I'm really very thrilled to be here with you this morning. For those of you who are coming from outside, very welcome. Welcome to Puerto Rico. And I have to tell you that you came at the right time and the right weather. And don't spend too much time in this meeting. Spend some time. It will be very important. But please enjoy this weekend. I'm a population geneticist, actually, in the Department of Biology at the University of Puerto Rico at Mayaüez. This is mainly an undergraduate institution, and we have a master's program, not a Ph.D. program. And we have over 1,600 students that are undergraduates, and we do most of our research with these undergraduates. And actually, I am, let's see if I can. Okay. So in our lab, we have this NSF grant called the Local Genome Diversity Studies, in which we have dozens of undergraduates collecting mouthwash samples all throughout Puerto Rico. Our aim is to have 96 samples from each one of the 78 Puerto Rican municipalities. So that would be over 7,000 samples, so that we have a DNA repository to carry out whatever studies need to be done. And so far, we have over 2,700 samples. And as you may see, the municipalities that are in red are those that are completed. Those that are in light red, they have been started already. The ones that you see here in light green have not been started yet. So you can see that, in fact, our students are mainly from the western part of Puerto Rico, and that reflects in the distribution of the samples that have been collected so far. Now, I am sharing my lab with Tarasso Lexic. He's actually the PI. I'm the co-PI of this grant. And the graduate students who are leading this work are Yashira and Israel. And up to last year, our main undergraduate was Maria Albertorio. She was my undergraduate, and she's now in Rochester University. We are very sad to have lost her. Well, she did a great job. And the study I'm going to show you now is that we took 192 of the samples that we have collected now, and we sent Yachira and Israel to the National Cancer Institute last summer so that they would actually use the ion AmpliSeq cancer primary pool to genotype passenger mutations in 192 samples. So we selected the samples from all over Puerto Rico. We selected 12 municipalities and sent 16 samples from each one of those 12 municipalities. And the ion ampli-C cancer primary pool is designed to use 190 amplicons. Actually, only 189 were applied, and 184 of them worked out. And the 190 amplicons interrogate 46 cancer genes. Even though we actually used only 184 of them, we still were able to interrogate at least part of these 46 cancer genes. In total, the 190 amplicons interrogate 744 non-passenger variants. Our 194 amplicons that we were actually able to use successfully interrogated 724 variants. And of our 192 samples, one actually dropped down. And we tested only 191 of them, but we got 166 of them that were of good enough quality for giving us some results. So, summing up, we applied 184 amplicons that interrogated 724 passenger mutations in 46 genes. 63 of the 724 mutations were positive in 160 samples, and they were distributed among 26 cancer years. So we got 20 cancer years that gave no positive results, and of the 26 that gave positive results, approximately we got of an average of approximately 3 passenger mutations per cancer gene. So these are the 46 genes that were interrogated. The ones that you see with the apostrophe are the ones that gave at least one positive result. And these are the site frequencies. So we have the variants that were positive ordered according to the genome position in the x-axis. And this is quite surprising the first time for a person like me that had never worked with cancer before. You find that, you got five variants that had a frequency of over 150 out of the 332 chromosomes that we looked at, right? So definitely we needed to see what these variants were. And there was another one here that that was found 70 times in our 332 chromosomes. Right? So I'm going to spend a few minutes talking about these variants. The first of the most common variants that I want to talk to you about is this one. It's in the APC gene. It is a synonymous mutation in a codon that codes for threonine at position 1493 in the APC. And the NCBI DBSNP database gives you some references you can look at for each one of the polymorphisms. And I did that for each one of these polymorphisms. And the most relevant paper I found among those for this polymorphism was a work done in Taiwan, in which they found that the alternate allele is found with a frequency of 82% in healthy people, but 93% in people with colorectal cancer. These are germline variants. And they actually found a statistically significant difference. So they associate the A allele to colorectal cancer, and the A allele is actually the most common, even in healthy people in Taiwan. And it's also the most common in general population in other continents. Like, for example, in CEU, it has a frequency of 64%. Among Yorubans, 53%. In Puerto Rico, it is 65%. And the distribution of this variant is shown here. It is most common in the central part of Puerto Rico and also in the northeast. Another one of the, actually it's quite low in Aguada here. Another one of the common variants is in the platelet-derived growth factor receptor alpha. It is also a synonymous mutation in a codon coding for proline in position 567 of this protein. And I found this paper that was done by a group in Portugal. And they did the work on 30 women with carcinoma of the uterine cervix, and they found that the RTN allele was found in 30% of them. However, when you look at the general population in Africa, the RTN allele is present between 80 to 90%, depending on the population you look at. In Europe and Asia, it's very high. It's up to 97%. And in Puerto Rico, it is 98%. So remember, this work was done in Portugal. So what we find is that the RTN allele is actually at a very low frequency compared with the general population in people with, in women with uterine cervix carcinoma. So we have to conclude that this variant is actually protective. It's actually a good thing to have it. So it's actually very nice news to have 98% frequency in Puerto Rico. And its frequency is, the one we found is 100% in most of the municipalities. They were lower in the east and also in the northwest of Puerto Rico. For the other variants, we found rather very weak or actually negative evidence. Remember, these are passive mutations. They do not necessarily are bad. One of them is in the epidermal growth factor receptor gene. It is also synonymous in a codon coding for glutamine at position 797 of this protein. Actually, in the same paper, it is found in the paper from Portugal. The derived allele was found in 56% of the patients of carcinoma for the uterine cervix. And the frequency of the alternate allele among Yorubans is 36%. In Europeans, it's 64%. So the difference is not really very high. And when you consider that the number of samples was really low, so this is really very weak evidence. In fact, it's not significant. And among Japanese and Chinese, the frequency is rather lower. And in Puerto Rico, it's 56%. So this is a very weak evidence, and my conclusion is that there is no strong evidence to worry about this variant, so I'm not going to pay any more attention to it. And in fact, the other two ones that are very common is pretty much the same story. One of them is the fibroblast growth factor receptor 3 gene. We found that this was also a synonymous substitution coding for threonine. And in the red gene, also a synonymous substitution coding for leucine. However, one of the papers for the red polymorphism actually gave a very strong association to Hirschsprung's disease. Okay, the GG genotype was strongly associated to this intestinal disorder. However, this is a cancer talk, right? So I'm not talking anymore about it. The frequencies of the alternate variants, 98% in Puerto Rico for the FGFR allele and 80%. So that actually means that the frequency of the G allele will be 20% in Puerto Rico. Also common, the one that shows up 70 times in Puerto Rico, it's in the PDGFRA gene. This is also a synonymous substitution, coding for a valine residue at position 874. And this variant is actually listed in the catalog of somatic mutations in cancer. In the general population, among Yorubans, its frequency is 35%. CEU is only 16%, and in Asian populations it's 20%. In Puerto Rico it is 21%, very similar to the Asian populations. And the distribution is actually much higher in Ponce and lower in the central parts of the island. So, you have noticed that I have talked already about 6 variants, and all 6 were synonymous. So at this point, if you are paying attention to this talk, you should be asking yourself whether I found only synonymous substitutions, right? And the answer is no. Of the 63 polymorphisms that we found in Puerto Rico, only 29 were synonymous, and 20 were non-synonymous. So if we get rid of the 6 that are most common, we have pretty much as many synonymous and non-synonymous substitutions. We also have 12 intron variants, 1 frame shift, and 1 variant in the 3-PAM UTR. So at this point, you have to start wondering why we find only synonymous substitutions among the variants that are most common. And if you think about it, you know that the non-synonymous substitutions usually have a higher impact on the expression of the function of the gene function, right? So it is quite likely that the reason they seem to be at a low frequency or non-existent in the high frequency variants is because they are actually deleterious, and they are just selected against, right? So taking this as an hypothesis, I started looking at their frequency according to the frequency of the variant. How many times the variant shows up in our samples. So I made the 4 categories, right? Those variants that show up between 10 to 326 times, those that show up between 3 and 6 times, those that appear twice, and those that were found only once in our sample. And how many of the variants fall in each one of these categories? So we will find that whereas only 16 appear between 10 and 326 times, and only 10 between 3 and 6 times, we have 27 that show only once. Right? So the biggest group is showing only once in the 166 samples. And you have to wonder, how are the non-synonymous substitutions distributed among these categories, and also the frame shift mutation. And it shows clearly that the ones that show up three times or more have a much lower frequency of non-synonymous and frame shift mutations than the ones that show up twice or less. Right? So clearly, the mutations that are a priori thought to be more of having more impact on the gene function are in fact showing with more frequency in the variants that show with a lower frequency. Right? So our conclusion is that the real bad ones are the ones with a least frequency, and those are the ones we should be paying attention to. So if those are the real bad ones, you will think, okay, then it should be cited in cancer studies. And I take this information also from the DB-SNP. However, that's not the case. When you look at them, of the 2 that are in the 10 to 326 categories, 1 has been cited. Of the 2 that showed up between 3 and 6 times, only 1 has been cited. But of the 17 that show up twice or less, only 2 have been cited. Right? So clearly, our work, the work that scientists have been doing so far, have concentrated in the mutations that are very common. Maybe because they show up all the time, right? But probably also because it's easier to work on them. The variants that are in a very low frequency, you need to work with very big samples to do any meaningful work on them, right? So that's very hard. So it is evident that in order to find the meaning and the impact that these mutations are having in our population, it's evident that we will need to do work with very big samples. And in fact, you can also look at the cosmic mutations. Those are listed in the catalog of somatic mutations in cancer, and it's pretty much the same story, right? Both that are found with a frequency of 10 to 326 are cosmic mutations, but when you look at the ones that show up with the least frequency, they also show up with the least frequency in the cosmic mutation. So, our conclusions are that the ones with the least frequency need to be studied more. So let's look at them. The variants that appear with a low frequency, that's 6 times or even less. And in this small table, we have the number of samples. Remember, we got 160 samples. Of those, 2 people actually had 3 variants that are low frequency variants. Right? We got 17 people who have 2 of the low frequency variants, 53 people with only 1 of the low frequency variants, and 94 people who have none of them. How are they distributed in Puerto Rico? Well, on average, each Puerto Rican has about half of these low frequency variants, and there is a lot. There seems to be a lot of variation from municipality to municipality, from 0.19 in Orocovis up to 0.87 in Añasco. So I told you I'm a population geneticist, so I needed to do a Hardy-Weinberg equilibrium test. You probably know that from our work with other ecologists, we have shown that Puerto Ricans marry depending on ancestry. Right? Puerto Ricans who have higher European ancestry tend to marry spouses with higher European ancestry, and Puerto Rican with higher African ancestry also would marry spouses with higher African ancestry, and even a Native American ancestry. People with higher Native American ancestry would tend to marry with people with higher Native American ancestry. So much for being race blind in Puerto Rico. But the thing is that, so you will expect that in Puerto Rico there should be a very strong deviation from Hardy-Weinberg equilibrium. In fact, when you look at ancestry informative markers, that's what you find. So I was really expecting to find the same with these passenger variants. But the real big surprise was there was nothing like that. It was actually the other way around. It really looks like we want to have a very nice equilibrium in Puerto Rico. You know, in order to have the higher statistical power, it's better to use alleles that have a frequency that is close to 50%. And of all our variants, the one with the frequency that was closer to 50% was this one. Okay, the G allele 147 times, the A allele 185 times, and when you do the chi-square, P is close to 0.9. And it's the same when others, and you use, doesn't matter. I did this test for 10 different variants. The 10 variants with the alleles most close to 50%, and the results were always the same. Right? So what this suggests is that this doesn't depend, or depends very little on ancestry. To finish, I want to pay some attention to those low frequency variants that actually something was said about them in the DB-SNP page that showed pathogenic clinical significance. We had 2 of those. One of them is in the serine threonine kinase 11 gene. This is non-synonymous mutation, and it was found once. It was in Rio Grande. And the other one was in the APC gene. It's also a non-synonymous mutation, and it was found once. It was in San Juan. Interestingly, we also found what seems to be a very strange frame shift, and a very serious one. There's no RS number for this variant. It is in the tumor suppressor gene P53, and it's in the 5th exon. That's fairly early, right? So it should be very detrimental, a very deleterious gene, and or variant. And it's actually found 4 times, and 3 times in Rio Grande, and 1 in San Juan. So there seems to be a cluster right there. Right? So I hope you enjoy my talk. So that's it. Thank you very much.
All right, so before moving on to our next speaker, let us all take a moment to thank Dr. Martínez-Cruzado for his talk. Gracias, doctor. Okay, so keeping in line with the previous presentation, we learned about how ancestry and polymorphisms may affect prostate cancer risk and induce a more aggressive disease in our population. Our next speaker, Dr. Margarita Ramírez Irizarry. She's a professor at the School of Health Professions in the University of Puerto Rico Medical Sciences campus. She's also a researcher in the field of prostate cancer and seeks to try to unravel the causes for the high mortality rate in men with prostate cancer in Puerto Rico. Her laboratory is doing research using both proteomics and genomics approaches to study prostate cancer. Please help me welcome Dr. Ramírez Irizarry. Margarita Ramírez
Irizarry-Ramírez.
Ay, perdón. Sorry, Irizarry-Ramírez.
Talking about ancestry. Bonjour, bon dia, good morning, buenos dias. I think that covers most of the attendees. And you have heard from the premier population geneticist in Puerto Rico, so I have a daunting task. And I knew it when I saw the program, and I said, oh my God, after Martínez-Cruzado, how am I going to do this? But anyway. So, as you can see from the first slide, our work includes the sponsorship of many institutions, and talking about transdisciplinary, as we were talking yesterday about transdisciplinary research teams, this is our research team. From the medical sciences campus, you can see that we have people who are, I am a biochemist and molecular biologist by training, by the way. And so I have clinicians who are my colleagues in this work. I have pathologists and a urologist. I also, of course, have my statisticians, Eric and Maribeliz, who work with us. We also have the collaboration of Dr. Sanchez-Ortiz, who is another urologist, which is not at the medical sciences campus, but has provided us with most of the samples that we have been testing. From the MD Anderson Cancer Center, my colleague there is Dr. Petaway, who is a neurologist. And then, of course, they also have other people in the statistics and pathologists and whatever. And then we have Dr. Rick Kittles, who have been doing most of the ancestry determination for our samples. So why are we studying prostate cancer? Well, to be honest, the first answer was because nobody else was doing a systematic study of prostate cancer in Puerto Rico. There were bits and pieces around, but nothing really that could be called systematic. So yesterday you saw some of these statistics. These come, of course, from the cancer statistics, and the references are right there. But anyway, what I wanted to show here, these are statistics for 2008, these are statistics for 2012, and as you can see, we are not doing much better in terms of estimated new cases. 2012, 241,000. This is for the US, and Puerto Rico is not included here, okay? And estimated deaths, again, is more or less flat. But what is the picture for Puerto Rico? The picture for Puerto Rico is that, as you can see, our incidence is not very high in terms of other, as compared, for instance, with the US Hispanics, or is lower than as compared with the US Caucasian. Of course, is lower still than the African American, but that is not surprising because the African American as an ethnic group is the worst faring group in terms of prostate cancer and in other cancers. But what struck us that being the incidence, then we had this mortality rate. And when you look at 28.3, it doesn't struck you maybe like such a high mortality rate. But then when you look at these maps, and this is the map from the cancer registry for 2000 to 2004, it struck us that even though that was the mortality and that was the incidence, when we look at the map, it was obvious from the colors, and orange is the higher incident and deep blue is the higher mortality rate, that the mortality rate and the incidence were not distributed equally in Puerto Rico. Moreover, when we look at the most recent, which were published last September, and this is from 2005 and 2009, again, we are seeing big pockets and small pockets, but big pockets of higher incidence, big pockets of higher mortality. And I know it's difficult because you just saw the other one, but it struck me when I saw this last September that now you can see almost like a corridor from the Arroyo area, Patillas, Maunabo, Yabucoa, so on and so forth. If you include this sort of turquoise color, which is the second highest mortality rate, and also, except for the Luquillo, it's almost a corridor of high mortality rates. And then our esteemed colleague had published in 2011 and I urge you to look for this paper. This is a very elegant work in which it's called The History Shaped the Geographic Distribution of Genomic Admixture on Puerto Rico. And then, if you remember the maps that I just showed you, and then you look at this, and then you look specifically at this area here, which is showing higher African ancestry. Then we were struck by not only because this is the higher African ancestry, and it looks higher African ancestry, higher mortality rate, higher Native American ancestry, lower mortality rates. So what is going on? Is there any relationship? Can we establish such a relationship? Could that help us explain what is going on with prostate cancer in Puerto Rico? So we posed a hypothesis and said that we thought that there could be a relationship. although he did not go into it, but he did mention them. So we have a powerful tool, which is called the Ancestry Informative Markers. And with those ancestry informative markers, which are sets of polymorphisms that appear with different frequencies between populations from different geographical regions of the world, and therefore can be used to estimate the geographical origins of the ancestors for an individual right now. So we have this type of tool. And because already some researchers had established that there were some specific areas of the genome that had polymorphisms associated with aggressive prostate cancer. And those areas of the genome associated with aggressive prostate cancer had been amply described for the African American population. We decided then to look if in Puerto Rico, West African ancestry was in any way associated with prostate cancer in this heterogeneous Puerto Rican population. So to do this, we had two specific aims. The first one was to assess the association of prostate cancer risk and aggressiveness with the genetic ancestry. And the second one was to assess the association of prostate cancer risk and aggressiveness with those SNPs associated to very aggressive prostate cancer already known for the African American. Now, this is a work in progress. So what I'm going to show you is not a work that has been finished. These tables that you're going to see are just about a little bit more than half of the samples that we already have collected, okay? So let me, for a brief but important section, tell you what the study methods were. The first one that we have to define is who are our controls. This is a case control study. And our controls were defined, for those of you who were here yesterday, I did ask the question, as healthy men who had less than 2.5 nanograms per mil of the prostate-specific antigen in their serum, and that had a digital rectal examination that was negative. For the Puerto Rican men who are in this population who are over 40, could I have a show of hands? How many of you have done a DRE? So that gives you an idea of how difficult it is to collect the samples for controls, okay? So what we have done, is that we have actually done clinics in which my clinicians, colleagues in this study, have actually done the DRE, and we have tested them for the PSA in order to enroll them as controls. Okay, so those are the controls. Cases are actually the paraffin blocks of radical prostatectomies, but the cases are described as men diagnosed with prostate cancer and with the pathological results after a radical prostatectomy. Aggressiveness was assessed by the usual clinical parameters, and remember that the aggressiveness here is being assessed in the paraffin blocks in the pathological specimen. So aggressiveness was assessed by the usual Gleason score and the pathology staging. And of course, we also had the results of the prostate-specific antigen levels, and we also knew what the treatment outcome for specific patient is right now, because some of these people are still alive, and we hope that they stay alive. So yesterday, we also heard about the complexities of dealing with multiple international studies and how it is very important to have sort of SOPs for each and every area of this study. And Gleason score is one of those things, as the same as pathology staging, that you have to have agreement between pathologists. So what we have done is to have both the person who is the pathology at the MD Anderson Cancer Center and the pathology here read what the original pathology read in the slides. So we had the results in paper, but we also asked for the slides. And then these two people who had already agreed upon what the parameters were to say that the Gleason was either 3 + 4 or 4 × 3 or whatever, they all read again the slides in order to minimize possible confusion in terms of this assessment. Then aggressiveness, as I said, was defined by the Gleason and also by the pathological stage, which here is a summary of the different subdivision of staging associated with prostate cancer, in which, of course, from better to worse, it goes from T1 to T4. But as you can see, there are subdivisions. And again, this is what the pathology looks at in terms of if it's prostate cancer that is still just in one section of the prostate, if it's a prostate cancer that had gone up to the outside capsule, if it's a prostate cancer that had invaded the lymphatic nodules, or is it a prostate cancer that already were even out of the nearby lymphatic nodules and was metastasized. Recruitment was done by age matching by groups, cases, and controls. We also wanted to see if we could have representation from different areas of Puerto Rico because of the maps that you have seen previously. So although we had samples from the medical center in San Juan, and for those of you who are not from Puerto Rico, I have to say that the medical center in San Juan, although it is in San Juan, is a tertiary clinical facility. Therefore, the medical center receives people from all over the island. So that's one of the areas that we tap into for recruitment. The other is a private facility, but also receives samples from all over the island. So although we cannot say that we really set out to say, well, we wanted, like Juan was saying, this specific number from each of the municipalities, that is not so. I mean, that's the optimum, but that is not so. But anyway, we do have nowadays a representation from almost all of the island. I think that the only places that we do not have samples from are Vieques and Culebra. Otherwise, we have samples. Now, in Puerto Rico, as you can see here, we have 300 cases, 300 controls. At MD Anderson, we have 200 cases and 200 controls, and we are comparing both samples. In their case, is only African American subjects, both cases and controls. In our case, is Puerto Ricans. And let me do another definition. Who is a Puerto Rican? So in terms of our specific study, a Puerto Rican was defined as someone who had parents and grandparents born in Puerto Rico. Or, in the case of grandparents, those grandparents that came from Europe, but mostly was born in Puerto Rico. You can do many definitions of Puerto Ricans, by the way, for those of you who are not from here. But we had to settle for some type of definition to be consistent. So that was our definition. Okay, so as I said, we then proceeded to extract DNA from paraffin-embedded tissue in the case of for cases, and from blood in the case of controls. And then we started doing the genetic analysis, which are going to establish first the correlations between the ancestry informative markers with the characteristics of the prostate cancer in the population, incidence, histologic features, prostate cancer, so on and so forth, and the associated SNPs to aggressiveness in the case of prostate cancer with the characteristic of each of the individuals in our sample. And then we added something which, on the way we recognized that we had to add, which was the effect that socioeconomic status may have in terms of defining the type of prostate cancer that these patients may have. So recognizing that incidence and mortality rates may be influenced by socioeconomic status, we set out to try and get as much of demographic information. And the type of information that we have gathered was, well, age was there, but then education. That has not been easy, believe it or not. People, for some reason, they don't give you education, and it's hard, it's hard. Also remember that our cases, we are not interviewing the cases. So we are relying on whatever information are in the medical charts. So our charts not necessarily have information on education, and not necessarily have information on income. So although those are two parameters that will have been very, very helpful to establish the socioeconomic status, in reality, we have gone in another way, which I'm going to show you in a little while, to be able to sort of, by proxy, establish what is the socioeconomic status of the samples. We also ask them, of course, the address, and we also ask them for familiar history, both cancer familiar history in general and prostate cancer family history. So the expected outcomes of this work is to see if we can make a correlation of the risk and aggressiveness of prostate cancer in our population related to the SNPs, and if those SNPs have differences in frequencies between the cases and controls, and also between the staging and the grades, and if the patient has recurrent disease or not. Remember that I told you that we know what the outcome for each specific individual is of the specific disease. We also, as I mentioned before, want to establish if there is a correlation with the West African ancestry, the socioeconomic position with the disease phenotype, and the SNPs. Will this, which is something that is always asked, will this study have any clinically significant outcome? Well, we do hope so if our hypothesis is correct, because it will help us in establishing some sort of future model for early detection, and also for prevention, and probably for prognosis. But we still do not have the answer. So preliminary results. As I said, it's preliminary. We are now here. This is 106 AIMS, which is the informative markers, and this is 664 DNA samples. We are now up already to about 800. But anyway, from this study of the ancestry, you can see Puerto Rico samples are the dark circles. African American samples are the clear circles. And the mean West African ancestry in Puerto Rico was estimated at 22%, just as Dr. Martínez-Cruzado was saying, and 73% in African Americans. And of course, Puerto Rico also had a higher Native American and European ancestry than the African Americans, which we sort of expected, what we have to confirm. Now, we set out to do some of the relationships already. And what we have found, for instance, is that, first of all, that we need more sample. Even when we get to our sample size, the 300 and the 200 and whatever, we already know that we need more samples. So that's our future work. We will continue to recruit both cases and controls. That's for sure. But anyway, as you can see here, this is cancer status. I mean, these are the cases, these are the controls. These are West African, European, and Native American for Puerto Ricans and African Americans. And right now, the only thing that we can say, the other thing is that we categorize the cases by Gleason score less than 7 and Gleason score more than 7, because in pathology, more than 7 mean more of an aggressive disease. Less than 7 in terms of the Gleason score is less aggressive disease. So to do some sort of a rough estimate of what we were looking at, we did this separation in 2 categories. And the only thing that we can say right now is that apparently in Puerto Ricans that have a higher West African ancestry, but I mean, you are seeing the numbers. There appears to be a group that have Gleason score that are higher than 7. But there's not enough sample here to do really a conclusion on that. So the rationale for the search of the SNPs associated to prostate cancer risk is, as Dr. Martínez-Cruzado was just saying a while ago, is that the risk allele or alleles will be in varying frequencies in the ancestral population, and that we will be comparing the admixture mapping between the cases and the control, and that this ancestry may fluctuate around the estimates that we are doing, but also that this mapping can identify regions where cases and controls differ in their underlying genetics, which may explain again what's going on with the disease. So these are SNPs that have already been identified as SNPs for prostate cancer risk. And again, this is West African, African American, American, European, and Asians. And there are some SNPs, as this one, for instance, and this one, for instance, that appear to be very well recognized in the West African ancestry and that appear in the African American population and have been associated and described as risk alleles. And they are in the region of chromosome 8, Q24. I showed you previously a diagram in which that region was sort of mapped. And it appears that although some of these SNPs do not, or we do not know what the actual function is, they are very near a region that has been associated to cancer for a long, long time, which is the c-MYC gene. So, you know, although some of them we don't know what they do, we know that it's an area associated with proliferation and cancer. Other studies have identified this group of this group of alleles, and again, HQ24 in chromosome 8, and some of these are associated with molecules like this one here and this one here, who are associated with molecules previously not related to cancer, which are, for instance, the interleukin-16, which we don't know what's going on, but is showing up in prostate cancer as something that may have some type of relevance. Again, this is another study. This is a completion of 4 studies. And again, you are seeing some of the, here is again the 1691, so on and so forth, the 117295, and so on and so forth, that keep up showing and showing in areas in the AQ24. So this is our results right now. And in these results, forget about this one because Hardy-Weinberg equilibrium here is completely off, and I think we have some issues there. So I wanted to put an arrow there because I knew people were going to see it and what's going on. So forget about that one. But nevertheless, we have this one here and this one here, who appear to have some sort of significance already in terms of the prostate cancer risk in our samples. This one may be approaching, but again, we do need more samples to really assess if there's a significance for that particular risk, allele. So we can say that we have now, out of that 15 SNP panel, we actually have 3 that appear to be possible candidates. Now, I have to tell you that if you go into the literature, you will see that this is a constantly evolving field. I don't know how many papers come out, and just recently, like 3 weeks ago, there's another paper coming out in terms of possible risk alleles for prostate cancer, possible aggressive alleles for prostate cancer. And again, it has to do with the diversity of the populations, the diversity of the environment, and so on and so forth. But anyway, right now in our study, we are identifying those possible 3, and we are testing for 15 other SNPs, by the way. But out of those that have been tested, 3 possible SNPs that may have, in the end, some sort of significance. So as I mentioned before, we set out to address the area of the socioeconomic status and prostate cancer because we wanted to be able to say, well, it is something that has to do with the genetics, it has something to do with the biochemistry, maybe what is the effect that the socioeconomic status has, because we know that even in Puerto Rico, for those of you who are not from here and that see the island as a very small island, the truth is that we know that there's not equal access to medical care throughout. So whenever you see, well, this person has a very high-grade prostate cancer, you may ask yourself, is it because from the onset was a very aggressive prostate cancer? Or is it because at the time that it was diagnosed was so far on because the person reached very late to the actual clinical care, and therefore it is not a matter of aggressiveness of disease, but it is a matter of clinical access. So then, Dr. Eric Suarez and Maribelis Soto has done an analysis in which, using principal component analysis and 4 socioeconomic indicators available in the US Census that are unemployment, annual household income, percentage population living below the poverty level, and so on and so forth, years of education, etc. They came up with a way of trying to establish the socioeconomic position as a proxy for socioeconomic status in this group. And after computing the socioeconomic position, they categorize our group in 5 categories, in which the socioeconomic group 1 represents the highest socioeconomic level, or as they say, lowest social deprivation. And it took me a long while to think about lowest social deprivation. And SEP 5, meaning the lowest socioeconomic level, but for them is the highest social deprivation. So every participant was identified with one category of the either 1, 2, 3, or 4, or 5. Now, we had another problem, which is how to really indicate where the cases and the controls are coming from. The controls are easier because you ask them the address. The cases is medical records. So the addresses in the medical records not necessarily are physical addresses. So what they did was to try and match the addresses that the cases had with actual census blocks. As you're going to see in the next slide, you're going to see that there's a column that says match, and there's a column that says unmatch. The unmatch are those cases that could not be assigned to any specific census block for different reasons. Addresses were incomplete, there were PO boxes or whatever, and the fact of the matter is that they could not be matched. Okay. And for the slide that is upcoming, we define again severity risk groups as low risk with a PT2 and Gleason score less than 7, etc. And high risk PT3, or post-positive surgical margin, Gleason scores greater than 7, or a combination of Gleason score in which it was a 7 but 4 plus 3, and not a 3 plus 4, because that has some implications in pathology. So here we have, as you see, the match and the unmatch. And out of the many relationships that they have established, it appears that right now this has a p-value that is statistically significant, and it appears that in the unmatched, we have differences in terms of low risk or high risk as compared to the match. Now, the question is, we were discussing this the other day, why? Well, maybe, and this is completely speculative. I have no empirical data whatsoever. As I mentioned, the unmatched are those addresses that we could not match because they were what? Probably rural areas. The addresses were PO boxes that didn't have to do anything with El Barrio, whatever, Sonadora, or Indiera Alta, or whatever it was. And maybe these are the people, again, who do not have access to medical care. But again, that's completely speculative. I'm finishing, don't worry. And the other thing that has come up already is that for those cases that are residing in areas of lower social deprivation, which is SEP 1, they seem to have a lower proportion of people, of men in the high risk category versus cases that are living in the highest social deprivation areas. However, this difference was still not significant. But again, as I said, this is why, since we have these tendencies, and this is just one example of what we had found, we need to add to the sample size to be able to really come up with significance in the analysis. So to conclude, we have to, right now in this work, finish the analysis of the controls, which we have not done, increase the number of samples, and then we have submitted another proposal to include other clinical parameters that might help us in really pinpointing the aggressiveness of the disease, and to assess in each case the status and relationship of the aims with the disease and the SNPs with the disease. So there's still a lot of work to be done. And finally, I want to acknowledge all the funding from NCI, from the National Institutes of Minority Health and Health Disparity, the Department of Pathology and the School of Health Professions from the University of Puerto Rico, the Veterans Administration Caribbean Health System, and the Robotic Urology and Oncology Institute of Puerto Rico. And we couldn't have done the work without all of these persons. Thank you.